Hello and welcome everyone. This is the second podcast for day 39, chapter 9 over respiration. In this podcast, we're going to focus on objective 3, more of the nitty-gritty behind the different stages of respiration. All right, well, let's go ahead and get started. All right, let's begin our discussion of cellular respiration. For our discussion purpose, I'm going to draw this large eukaryotic cell. And remember, respiration also occurs in aerobic bacterium, but for example here, I'm going to use a eukaryotic cell. And because of that, I'm going to draw a very large mitochondria, disproportionately large to the size of the cell. And I'm going to draw our cristae here, our inner membrane. Just to remind you, this is our outer membrane. This is our inner membrane, and this is our matrix. I may erase that in a moment because I may need the space, but that's our matrix. Your book describes 4 stages of cellular respiration, so I'm going to talk about them as 4 stages. The first stage is glycolysis. Glycolysis takes our sugar, our glucose, and it converts it to pyruvate. P-Y-R-U-V-A-T-E. It makes 2 of these, in fact. If you ever read somewhere where it says pyruvic acid, it's the same thing. Just 2 different ways to say the same thing. This glucose comes into the cell from outside. Maybe it's something that this eukaryotic organism ingested and now it's inside the cell. The pyruvate now will go inside the matrix. Inside the matrix, this pyruvate, and I'm going to abbreviate this as just PYR, and it's taken into, and it's transformed into acetyl-CoA A. And this is our second step of cellular respiration. And we call this pyruvate oxidation. I'm going to erase matrix because I need that space now. This acetyl coenzyme A now is taken into our third stage, which is called the citric acid cycle. I'm going to abbreviate that. C-A-C. Citric acid cycle. And it takes this acetyl coenzyme A and it makes a couple things, but primarily what we're interested in is it makes this NADH and FADH2. And these are essentially electron carriers. And it takes these electrons into our fourth stage. I'm going to write it here, 4, which is oxidative phosphorylation. And that occurs on the inner membrane of the mitochondrion. So I'm going to go over this again, and I'm going to add a couple more things to it as we go over it. So the first stage is glycolysis. It takes glucose into pyruvate. I didn't mention it before, but it also makes some ATP. Not a lot, but it does make some. This pyruvate is now taken into the mitochondria, into the matrix of the mitochondria, where it is converted into acetyl coenzyme A, acetyl-CoA. And we call this stage pyruvate oxidation. This acetyl-CoA is now taken into a citric acid cycle. This citric acid cycle, its main claim to fame is that it makes NADH and FADH2, which are electron carriers. It also makes some ATP. Not a lot, but it does make some. These electron carriers here are then taken to the inner membrane where oxidative phosphorylation occurs. During oxidative phosphorylation, it is safe to assume that we make a lot of ATP. Alright, I'm going to erase this and go over some other points from this particular objective. Alright, I want to make sure we describe exactly what NAD plus and NADH are. I mentioned it a little bit on the last slide, but I want to make sure we talk about them a little bit here. Alright, I don't want you to really memorize what the long names of these are. You can look that up in your book. It's nicotinamide adenine dinucleotide. We're not going to ask you that. But what you should know is that NAD plus is an electron carrier. It accepts electrons. And this is really important. You should also know that this is the oxidized state. NADH is the form of the molecule that has the electron in it, in the form of this hydrogen molecule. And it is the reduced state. And it's fairly easy for these two molecules, NAD plus and NADH, to go back and forth. It is this reduced form that is critical in delivering delivering electrons for oxidative phosphorylation. Okay, now I'm going to talk about each of these four stages: glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, and summarize what their overall purposes are, what gets put inside of that reaction, and what comes out. So let's start with glycolysis. Alright, so let's summarize it and say we take this glucose molecule. This is our input. This is what's put into glycolysis. And our output is 2-pyruvate plus a small amount of ATP and NADH. Again, our electron carrier. This is our output. The purpose of glycolysis is to break down glucose to produce these molecules. We should also remind you where the location is. This occurs in the cytoplasm. Next, let's look at pyruvate oxidation. Pyruvate oxidation takes pyruvate. This is our input, and it converts it into acetyl-CoA. That's our output. This occurs in the matrix of the mitochondria in eukaryotes. In bacterium, it occurs in the cytoplasm in aerobic bacterium. The purpose of pyruvate oxidation really is to solely make acetyl-CoA, because acetyl-CoA is needed for the next step, the citric acid cycle. This is what we're going to call it for our course. You may have heard it called the TCA cycle or the Krebs cycle, but the better term for it is citric acid cycle. Acetyl-CoA, this is our input. This is what gets put into this cycle. And as the name implies, this is a cyclical reactions that occur, and it produces CO2, ATP, NADH, and FADH2. Remember, these two are electron carriers. This is a byproduct, which will be released back into the environment, and ATP can be used as energy. This occurs in the matrix. And again, we could say in eukaryotes, and then in bacterium, it would be in the cytoplasm. in aerobic bacterium. Alright, let's move to our fourth cycle, our fourth stage. And our fourth stage is oxidative phosphorylation. You may have heard of this as the electron transport chain. They're the same thing. Okay, so the main input for oxidative phosphorylation are electrons. And these electrons come in the form of NADH or FADH2. These are our electron carriers. In addition, an input is oxygen, and we haven't talked about that yet, but we will. These are then converted through oxidative phosphorylation into our output, which is ATP plus water. So what is oxygen doing here? Let's talk about that for a little bit. Oxygen is the final electron acceptor. It accepts the electrons and is then converted into water. This water here. It does it by this enzyme, and I'm going to write it just so you have it in case you want to write it down. We won't ask this on the test, so if you don't want to write it, you don't have to. But I'm going to talk a little bit about it. It's cytochrome C oxidase. Cytochrome C oxidase. The reason I want to talk about this for a little bit is to try to convey to you the importance of oxygen in cellular respiration. This is the only part of cellular respiration where we need oxygen. But it's a very important part, because if we inhibit this process, it can kill the cell and ultimately kill the whole organism. So I'm going to erase this here just so I have a little room to draw here. And if you think about this poison called cyanide, or even carbon monoxide, they work very similarly. What they do is they inhibit this cytochrome oxidase. And so now cytochrome oxidase cannot act as this final electron acceptor and convert it into water. And by inhibiting this enzyme, what essentially happens is that the cell suffocates because it's not able to use this last oxygen. Much in the same way you think of being unable to breathe in oxygen would kill you. By blocking this enzyme, even though you can breathe in oxygen, you're unable to use it. So it's the same thing. So each of the cells now undergo kind of a cellular suffocation. So by serving as this final electron acceptor, oxygen plays a critical role in the overall cellular respiration, going from glucose to ATP. As we've been talking about the breaking down of glucose into ATP, I've mentioned that ATP is made at various steps. And so I think it's appropriate to talk about that just real briefly. It won't take too long. So we have glycolysis, we have the citric acid cycle, and then we have oxidative phosphorylation. One molecule of glucose will make 2 ATP during glycolysis. It will make 2 ATP during the citric acid cycle. And finally, it will make 26 or 28 ATP molecules during oxidative phosphorylation. So in total, from one molecule of glucose, 30 to 32 molecules of ATP can be generated. You'll see in some other textbooks and as you go into biochemistry classes that this number may change some. There are many things that can influence this pathway that can alter these numbers, but for our class, these are the numbers we will focus on. Now for the last question in this objective, the question asks, can cells break down other things besides glucose? And the answer is yes. So let's just kind of talk a little bit about this. We can have proteins broken down. We can break down carbohydrates. And we can break down fats. So you can look at this figure as a reference, but the main point we'd like to make here is that regardless of which organic molecule is broken down, proteins, carbohydrates, or fats, they all get fed into one of the three oxidative phosphorylation pathways. So they may get fed into glycolysis, they may get fed into the citric acid cycle, or they may get fed into this short pathway here, the oxidation of acetyl-CoA. So proteins can get taken into glycolysis, they can get taken into the oxidative step of phosphorylation, or into citric acid cycle. And it does it because the proteins are broken down by various pathways, reactions that we're not going to learn here. You may learn some of them in biochemistry. But these are pretty elaborate biochemical pathways that do this. But it can happen. And same thing with fats. Fats can also enter into glycolysis, or they can enter into the acetyl coenzyme A oxidative step. Ultimately, wherever they enter at, their goal is to get into this electron transport chain. Or I should call it what your book calls it, oxidative phosphorylation. So the answer to that question is a resounding yes. Many different kinds of molecules are broken down and converted to energy, but they do it all in a similar pathway function by entering into one of these steps of cellular respiration. Okay, that ends this second podcast. We'll have one more podcast over Objective 4. It should be a pretty short podcast. Let us know if you have any questions. We'll see you in class. Bye.